Hello? You're muted.
Yeah, I am muted.
Yeah.
Hello.
Hello, how are you?
Not bad.
That's good.
Tired.
Yeah, I imagine. Okay. I hope other people show up. Jesse said he probably won't be able to make it. Here's Ankit, little Ankit. Jesse said he said he's pressed for time, and
Sure.
so. But yeah, I think this was a great week. Very busy. We had the main conference, which I'll go over, and then also the Neuromatch, which I'll also go over. And nice to see people at Neuromatch. A lot of people at Neuromatch from the group, Ankit and Jesse and Daniela and Jenny, and I don't know, probably other people. And, you know, this is one of these things. We have the shared notes document, which I'll go over. But I think both of those conferences are on YouTube. So if you search for the YouTube channel, you can catch some of the talks. Yeah. And then, okay, for less time, that's fine. Is there anything you wanted to mention, Ankit, at the front of the meeting, at the beginning?
So even I've been writing like a blog or something related to the flash talks that I found was interesting. So when I have that done, I might like publish it. So like you need me to add me to the OREL publication.
Okay. I think you have to, I don't know if you have to request to join or if I have to send you an invite. I'll look into it.
Yeah. Yes. Neuromatch, I attended Neuromatch. So that was good.
Yeah. Yeah, there's a lot of content there. And yeah, it looks, I like that idea. Thank you for going through the flash talks and preparing something on that. I'll show in a minute that I prepared. I did something on like the general lab contribution. So we'll have those things up on the Medium. Just making a note to myself to invite you. I'll look into how, I don't exactly know how to do it, but it shouldn't be too hard. And then you can publish to the Medium through just the regular Medium. You have to be careful when you do it. There's like, make sure that you're posting under the right alias, because sometimes it posts under your own alias and then you don't know where it went. And so I've had that problem before. But anyways, so good. That's great. I don't know. I want to start. I don't want to start with the Slack channel first this week. I think I'm going to go and do it last, just because I want to see what it's like. So I'm going to start off by going through a couple things. Let's see. I'm going to start off by talking a little bit about this week and some of the things that from follow-up from that. So the first thing is this post that I prepared on the main and Neuromatch conference presentations. And I cross-posted this to the Medium. So if you go to the Medium, there's a short post there that links to this post. Just kind of like discusses like kind of where we were. And then Ankit can post his longer post. And then I can actually link that post to the short post I have on the Medium now. So, you know, it'll be people will come in and say, oh yeah, you guys did this, and then there's these flash talks at Neuromatch. So it kind of goes through the main conference. This is Jesse's tweet of our main conference submission. This is the actual submission. This is in the abstract book. I think I posted in the Slack about the abstract book. It's you go to the main website and there's a, like it says abstract book, you download it, and it's like abstract number 4 or something like that. And so it has like this stuff in it. It has a video, it has this graphical abstract, and then the written abstract. So that's the submission. And this is something that we've been working on for a couple years. We have 10 contributors on this, you know, ranging from myself to people like Rashab and Stefan Varetsky, who, there are a couple people in here who are GSOC students, a couple people in here who have been in the lab via Neuromatch, and then, so they've been involved in different levels of contribution. So this kind of goes through some of our preprints. And then this last part up here is something that we've been working on kind of more loosely over the last year. So this is the next step in this process, is to work on this inheritance and learning paper in like an agent-based environment. So, if you have ideas for that, maybe, we could talk about that as well. But that's the next step for this. So we have preprints now for the Gibsonian information, contextual embodied or continual embodied learning, metabrains, and this other paper that was kind of a metabrain example, and then inheritance and learning, which is coming up, and then developmental brain work vehicles, of course. So that was at Maine conference, and I'll talk about what went on there later. We also had a presence at Neuromatch 4, so Maine was on Monday and Tuesday, and then Neuromatch 4 was Wednesday and Thursday. So it was like just kind of switched over to Neuromatch. And I saw a lot of people from Maine at Neuromatch. Some of the keynote speakers, they were at Maine and vice versa. So there's a lot of crossover there. But we had 4 flash talks on 4 different topics. So this is the tweet that went out about this. It just kind of like, I tweeted for each abstract a single tweet, and then I wrapped it up in this one tweet where I put the links to those videos and materials, and then this is the logo. So this is pinned on the Twitter page for the lab, and so this is nice presence. And of course we were in the Discord, so I know Ankit was in the Discord, and Jesse was in the Discord, and Daniel was in the Discord, and we were talking a little bit about some of this. So each of these talks were 1.5 minutes long, and each of them had a little Discord stub. And I didn't see much conversation about the papers or the presentations, but this is something I think that's going to be around for a while, so people can come through. If they're in the Discord, there's a link to the Discord in the abstract browser. People will come back and look at it, and maybe we'll have some conversation later. But this is also something that's on, I know that we have 2 of them on the lab YouTube channel, and I know Jesse has the talk on allostatic kinds on his YouTube channel, and then I don't know where Daniel hosted hers. I think it's they're all, I think, in the Neuromatch YouTube channel, so you can go back to that. So yeah, we had four presentations. We had the universal theory of switching, which was this switching phenomena. This is something we're going to be working on more with the paper with Ankit, and then also some other things with Morgan, and is also interested in some of these things. So the paper that I'm working with Ankit is more of a sort of a technical paper for something like Gecko, but I'm going to be working on it over the next month, I think, and we'll be going over maybe how to partition this out. So I want to give I want to give people an opportunity to contribute to this. I don't want it just to be a technical endeavor, because the talk was pretty broad. So I want to be able to if we do 2 papers, one that's technical and one that's has other things in it. When I say technical, I mean algorithmic, not necessarily like.
Yeah.
So.
Yeah, I agree. Even I was thinking about that. Like the one we are working on was a little bit technical, and it does not have like the aspects from switching. So like switching could itself be another paper, and like that could probably be published in probably somewhat, let's say, offsite conferences maybe, or the
Yeah.
neurosciences conference. It's better for that, I think.
Yeah, I think so.
And that is like pretty like broad, and it's somehow like related to the kind of papers you write. So I think wouldn't be really difficult for you
Yeah.
because you're used to writing such kind of papers which are non-technical, describe the idea over a overview type. So that could be something like everyone could actually contribute to.
Yeah. Yeah. And I know that the CogSci conference is coming, like the CogSci conference is next summer, but the submissions are coming up like in the spring, so after the first of the year. There are also other conferences like A Life, which is in the spring, which, the submission deadline is early, and they want a full paper, so we have to have a full paper for that. So,
Uh, A Life, can you share the links of it?
oh yeah, I don't think they have their websites up yet, but one's like the annual CogSci conference, which is just CogSci 2022. I don't think they have a website up yet, but and then
So, so which one do you think we should target? It should be related to our topic, right?
yeah, well, a lot of these conferences are pretty selective, so and this includes Gecko. So we might want to say, well, we'll create a paper and then we'll just submit it and see what happens. And if it gets rejected, which is possible or probable,
Okay, right.
right, then we can put it somewhere else, but like we have the paper ready to go. So,
Yeah.
yeah.
There's a computational cognitive science meeting too, right?
Yeah, I think CNS.
Well, CNS is cognitive neurosciences,
Okay.
right? So cognitive
Yeah.
neuroscience societies, then that's in the spring.
Yeah.
I want to say there's a computational cognitive science, or and I was just wondering if you've thought about that as a target, just because you have a real computational program.
Yeah. I can't remember the name of it.
Let me.
Cosine, isn't it? Is that what you're talking about?
Well, there's cosine too, for sure, right? I mean that submission's already.
Yeah.
That's because that's going to be like February, because that's like NeuroPS. That's a skiing conference, right?
Yeah. Yeah.
So, yeah. It's like always. Is it always in Colorado now?
It used to be in Utah, and then I think they had it in Denver.
It's all ski resorts, right?
Yeah.
And yeah, so it's pushed out later because of the lack of snow. Yeah, here, I'll look for that. Sorry,
Okay.
but I think that might be good, just as
yeah, we'll have to. Yeah, I think finding the right venue is good, and then like I said, if we can shop it around to the different venues and just submit and see, and so, but we have to work on the papers now to get that in shape for the spring, because you don't want to do a crunch time. I'm not a big fan of crunch time work because you often just kind of, yeah. But yeah, and then you always end up kind of working hard towards the end to tie up the loose ends. But that's what you want to leave that time for, is loose ends. Like, did I get everything right in my figures? Did I, is there something missing in the text? Now let's do some algorithms like a week before the deadline.
So there is the ccneuro.org.
Okay.
Is the conference on cognitive computational neuroscience. And that is in the fall.
Right.
It's not exactly what. Anyway,
Okay.
it's especially if you've got computational work that's like genetic algorithms for behavioral modeling or something like that. That sounds like it would be very very appropriate.
Yeah, that's good. All right, thanks for that. Hello, Arthur. How are you? Glad you could make it.
I'm fine. And you?
Good.
Good.
Fine.
Did you have any updates for us this week or anything you wanted to mention?
No, I think that I don't have. I am just learning on how the dogs are working, and I am reading some articles to put in the brain organized on Jackman. So I am reading and trying to find the best one, and you can talk.
Yeah.
That's great.
Yeah, and I think this. I am I'm very busy with the university because now we have a lot of exams this week,
Yeah.
and but I am doing, I'm going and this.
Yeah, that's great. Thank you. Yeah, so I know there was a little conversation in the Slack about like doing some sort of like review or some sort of I can't remember what we said.
But mainly just a better job, like it's one thing to use Slack to kind of drop notes and to drop links and things, but at some point we really need to collate all this and get it into a doc, and just following. I mean, one, the Turing Way is already a nice book about data science, best practices, but it's a lot of people who are drawing from open life sciences already, and so a lot of their examples are actually very relevant for the brain organoids book, or like in the sense that there's we, in addition to the kind of material that we would be generating, we want a reference that gives people links to the various online repositories of knowledge databases and pathway databases, metabolite databases, and all the different knowledge sources that we would be drawing on to flesh out these, and you see this, and it's great. I'm glad somebody else is catching tissue talks,
Yeah.
where you can really see the level of detail that that these tissue engineers and tissue engineering scientists have on their they're it's really bottom up kind of work. Super impressive, and when they're looking at something like a kidney or or something where there's a relatively restricted range of cell types, where they've got these diagrams of like, we're tracking these 20 cell types, and when you think of the work from the Human Brain Project and Big Brain in particular, right? So this is this micron level view of the brain that was recently released from the European Human Brain Project in Munich. The amount of cell types, the cell heterogeneity, and is like astronomical, right? And it's it will be beyond people's ability to kind of track in their memory. So these will obviously be things that we're doing via databases and other repositories, and yeah. So that was also the link to another great EPFL Blue Brain data portal, was this neuroglia vasculature portal that we dropped in.
Yeah. Okay. Yeah, I mean, there's actually some talk at Neuromatch. There was a session on like, why is it that we have all these data that we're getting? People, is said in neuroscience, well, we don't have the data to make theories or to make predictive statements. But now we're getting it, and it's like, what do we do? And the cautionary tale there is, of course, the Human Genome Project. Well, I don't think it's cautionary, but it's basically that you've had the draft human genome for about 20 years, and we know where things are in the genome, but we don't necessarily know the function. And, that's a thing. Like, you can have these resources, but then taking the next step is often very hard. But it's necessary, and it's...
For sure.
Yeah.
And this is why synthetic biology is not an engineering program, but a scientific program, right? In that because one of the DNA conferences that I like to track is GP Wright, okay, like GPWRIT.
ATE, yes.
Yeah, and for this reason, right, is that it's a synthetic genetic programming conference, where sort of like the Ventnor Institutes, where developing a synthetic cell is both an engineering program and a scientific endeavor, right? In the sense that, like, you know more about what is needed and how the complex system works by designing your own cell, that a cell that doesn't exist in nature,
right?
But maintains, but is cybernetic in the sense that, like, it can maintain, regulate multiple systems within the cell and do the normal functioning, or the kinds of things. This is what we're working out, right? What are the kind of what's the minimum viable product for a synthetic cell that can maintain homeostasis and on a variety of metrics and can then undergo mitosis or meiosis, depending on what you've created.
Yeah.
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 And Blue Brain, I mean, this was like kind of, both the criticism and it sounds like, will also be the guide, right? That Blue Brain Project tried to do some of these things with the data that it had, but people found the products to be rather they found the products to be rather hard to understand. Right? I mean, you've got this famous or a paper that made headlines, but it made headlines sort of in the wrong way, where it's like, brain scientists say that the brain is 11-dimensional.
Right.
And not everybody really knows what to do with that.
Right.
But they were speaking very much about, like, on the basis of micron level cell typing and connectivity data.
Yeah.
For what kind of geometric structures and connectivity networks had been created.
Yeah. Yeah, it's good. Ankit had mentioned that he actually asked, does Maine have recordings? Yeah, so Maine has a YouTube channel. I don't know if they're up yet. Actually, if you're on Crowdcast too, you can join Maine. I think you can still join it. And they have the recordings there too.
Replay automatically.
Yeah, replay them. I like Crowdcast, but it's there's a cost to it, so we don't use it here. But Neuromatch has used it a lot. And, it's just if you go to crowdcast.io, and this is just, you just log in and you can search for, like, Maine, or, it's like you're in there.
If you've signed up, or if you've gone through one of their links already, it should show up in there.
Yeah.
Even if you didn't catch it live, it'll be in your login.
And then you said, yeah, I might start exploring comp neuro at the same time. I want to join in on proposed switching projects. So yeah, and neural decoding as well. So yeah, we'll talk more about that. Yeah.
Yeah, comp neuro is in the summer, and there's still time.
Yeah.
That'd be great.
Yeah. So let me go back. I know this was I kind of cut off in the middle of this blog post, but that's okay. We'll just, so that we talked about the switching paper, and then we talked about allostatic kinds, which is this thing that I've been working on with Jesse and Jenny and Avery. And this was I think that Jesse did a nice job of distilling this down to 7.5 minutes, because it was very difficult to kind of get through in 7.5 minutes, because you have it's very philosophical. And so, but I think this is a good practice if you are interested in doing this, like, working on your presentation skills. I would suggest doing something like this, submitting something to, say, like Neuromatch as a flash talk, getting 7.5 minutes, putting the slides together, and then just really kind of distilling it down to the basic stuff, and then sitting there and recording a talk where you talk for, you time it, you have your stopwatch, you time it out, you maybe do a couple takes where you go through, you develop a cadence, and then you can get it in the required time. It's a very useful skill to have when you do presentations, because oftentimes you'll have a time limit and you want to get the main ideas through. So to do that, you have to really be efficient. And so, and then Daniela did her talk on this economic metabrains idea. So she did a, I don't know if she has it available as a preprint, but she's done this paper on economic metabrains. So we did this paper on metabrains, and I've talked about it in the meeting previously. This is the thing Jesse and I have been working on with these layered artificial models of the nervous system, where you have different layers of representation, and there's like this geometric complexity. So you can have things that fold over, and then there are, connections between them, and there's the idea is that it enables you to do certain things in terms of processing. And so she developed this idea called economic metabrains. So she has these bioeconomic agents. They have a biological level, which operates at the level of the free energy principle. And then there's this economic agent, this rational agent, that has a higher level of representation that is so they work together. And this, I like the way it's set up. I don't know if she has if you watch the video, it's hard to follow, but again, it's like allostatic kinds. There's a lot of stuff in there. You have to read the preprint. And so, I mean, a lot of these talks are hard to follow if you just watch the talk. That's why I wish we had had a switching paper before this, but it's okay. But yeah, so I'll find out about that. And then finally, Gibsonian information, we did have a preprint. And this is exactly why I wanted a preprint for this, because we've done a couple presentations on this, but we haven't had a preprint. And people were, kind of like, well, I don't really understand what's going on. So, I think the preprint helps. So that's good. And this is, again, this work on thinking about information in terms of direct perception, Gibsonian principles from J.J. Gibson, who did this work on, information being direct perception, that you don't necessarily need a lot of representation in the brain. You just have a lot of sensory information, and you use that in ways that is highly informative. And this is a sort of a graphical abstract showing sort of, this is environmental information, very abstracted, and that you have these points and these flows. This is visual flow or sensory flow coming towards the agent. Here's this flow of time. So the agent is sampling things in time, and time is spatially distinct, because you encounter the things closest to you first and then farther out. Or if they're flowing towards you, the things that are closest to you, you encounter them first, and then the things farther away you encounter later. So this is a temporal process. You know, things kind of flow in time. And so there's this, we can characterize this using what we call Poisson distribution, which is this kind of distribution that has a long tail. And, you know, it counts the number of arrivals of something at a certain point in time. So you see that you have, you know, at this point in time here, you have 4 points. At this point in time, you have 6 points. And then you derive some descriptive statistics from the distribution. So your mean would be like, it would be, you know, there's a parameter lambda, which is like the median of that distribution. And that would be like really low in this case. If the lambda is higher, that means you have more things going on in the environment, the more cluttered environment. So there are ways that we can work on that. I put some math in the paper, but I didn't think it was highly interpretable. So I did this figure for it. So these are the kinds of things I think we should try to do for papers and for talks, you know, having things like this where we can kind of describe things a little bit more efficiently, you know, as we're presenting them or as we're talking about them. So that's all. And then this is linked to the Medium, which has, you know, there's a shorter thing in the Medium. Okay, so thank you, Ankit, for attending. He had to leave. But there's a shorter thing in the Medium where it links to this, and then I'll link to Ankit's description of the Flash Talks when he does that, when he publishes that. So the next thing I want to talk about is this. This is a blog post that I became aware of yesterday. This is on Pierre-Yves Zodier, who's from France, and he does a lot of work on what he calls developmental AI. So he's doing kind of what we're thinking of in terms of developmental work and developmental psychology, developmental AI systems. And this was like an interview that he did with someone on, it's called Intelligent Behavior Depends on the Ecological Niche, Scaling Up to AI to Human-like Intelligence in Sociocultural Environments. So he kind of goes through some of this, and he's advocating for a lot of developmental approaches to this. So there's actually a paper here, Intelligent Behavior Depends on the Ecological Niche, and I think this is in a German journal. I don't know if this is going to be in English or not. Well, this is where the interview was published. So it's probably, I don't know if it's in German or what, but this is like where it was published. But it's here now, so we can read it. So some of the questions they go through, is it useful to talk about strong and weak AI? What about AGI? And so Pierre says, you know, he's not a very big fan of this strong versus weak AI concept. When they speak of weak AI, they refer to techniques allowing to detect shallow relations between a number of quantities and used to predict what will happen in the future without really understanding what's happening. So sometimes people call it strong AI. That's a sort of an imaginary construct of AI that's a bit like AGI. So strong AI is like, this is strong AI. They don't really define it in terms of benchmarks or anything. It's just kind of like this fuzzy distinction between like, I can make something that looks cool versus like something that has a lot of, you know, sort of we can do a lot of benchmarks on, but it's like, you know, what's that distinction? I think that's a good way to think about it. And I think the farther we get into this developmental perspective, the easier it is to see how, you know, maybe those distinctions are false. You know, maybe those are products of sort of the way we kind of think about AI in terms of, you know, pattern recognition and things like that. So it's worth thinking about. And so then the next one is human-like intelligence is shaped by its sociocultural environment. So we've definitely talked about this in the group. We've talked about, you know, how, you know, you need to include things like culture and, you know, collective behavior. And I think people have, you know, kind of done this in terms of simulation, but they've not really thought about it in terms of the implications or sort of how that builds to like an intelligent system. So, you know, a lot of people have done this with ants, sometimes with human agents, but, you know, to kind of distill the sort of principles for So sort of like an AI, a set of AI agents. What does that mean? And so he talks about cultural evolution in here, which I like. And this is, of course, the next preprint that we want to do on this, on the developmental AI side, that we want to take some of the stuff we've done on cultural evolution and sort of apply it to agents. So, yeah, so ants are different than humans. Ants have a rich social environment, but it's not constantly changing. There's no cultural evolution in ants. This is causing a special family of problems that humans need to solve. So developing humans are always solving problems. Their environment's always changing. We do have things like ant colony optimization and swarm intelligence, but those are based on systems that are like ant and bee colonies, or termite mounds, where those things don't necessarily change. And I don't know if that's really true, but they basically have, they use a set of hormonal signaling mechanisms to communicate. They use, they have a lot of what you might call hard-coded or hardwired behaviors that, they're genes that are expressed, and it'll change their caste, for example, the social group that they're in, or it'll change their behavior quite directly. So these are things that are different from humans.
It would be, I don't know enough about it, but I'm interested to know what happens in ant wars.
Oh.
Right? I mean, that would be a change in environment, right? I mean,
yeah.
And there's this very, I've heard that there's these two species that are battling across California.
Oh, really?
Right. Yeah. There's these two kind of like, I don't know if they call them super colonies.
Yeah, I think so. Yeah.
Yeah, and there's a massive ant war playing out from like Mexico all the way up to Northern California. But I wonder what happened. I mean, there must be changes that to take place, even if those are still stereotypes, or like,
yeah.
Curious about that. I get that maybe they don't have culture, but
yeah.
they must have some other, and what happens and what breaks down
right
in those scenarios.
Yeah, I think that's that statement might be a little bit too provocative. I don't know, but I think the idea is that they're in a lot of the collective behavior in insects. It's more like about how they're hardwired in certain ways, like and they'll engage in behaviors like milling, where they sort of get locked into this behavior which they can't escape, and then they end up killing off the colony because it's like this feedback loop, the runaway feedback loop that they can't control. Of course, that happens in human societies too, right? We have all sorts of things that, catastrophes or wars that, we don't. But that's, that's something that I think is a debate in and of itself. I don't know if, but I think I don't think, well, people aren't thinking about that in artificial intelligence, or in if you go to even Neuromatch. I don't think people really are familiar with that, or I didn't see any talks on that. But, if you're talking in the A-life community, in artificial life, there are people who think about that. There are people who do swarm intelligence and things like that from an A-life perspective. And that's where you can get, and then of course you have cultural evolution as well represented there. So that's where you can kind of get that intersection. But still, I haven't seen too many people think about it in that way, where you're teaching some conspecific something. It could be parent to child. It could be, cohort to cohort member. And, it's passing down. People have these very specific definitions of culture and cultural transmission. So it's sometimes, people say, well, something is not collective behavior. It's cumulative behavior, cumulative culture. So it's you're accumulating knowledge. And in a sense, a termite mound or an ant colony is like that. You're accumulating information and acting on it. But that's not culture. That's something that's, different. Academically, anyways. I don't know practically what the huge difference is, but yeah. So, I mean, these are all these sorts of things are things to think about, like in terms of our own work, but also just kind of a different perspective on this. So, it talks about here about things like, tool use and making artifacts. That's another thing that distinguishes culture. Like crows make things, they make tools. Non-human primates make tools. Do they have culture? I mean, people would say they may have a rudimentary culture, but again, it's always that distinction in the academic sense. And so, yeah, this is, and then embodiment, of course, we get into this. Again, in our talk at Maine, we, champion the embodiment approach. And I saw there were a couple talks at Maine where they talked about embodiment. And, it's like, this is necessary for cognition and intelligent behavior. Not everyone said that. There were a lot of talks where they didn't think anything about embodiment. But, you still have to make the case for this. And then they bring up the example of a chatbot. And so chatbots don't have a body. It has actions and perceptions, but does it need a body to be intelligent? What is the minimum? And so this, I think, is a good question because, the argument for embodiment is that it adds to intelligence. And it may very well add to intelligence, depending on what you want to do. So if you want to just carry on a conversation, you need to be embodied. Do you need to have a body that interacts with the environment? Or do you just need to have like a, be able to input words and understand what they mean? I don't know where I come down on this. I think you maybe do have to have a body to really understand language, because language is more than just words. It's concepts. It's interacting with the world. And if you don't, know, like if I say the word fork, do I mean a utensil that you eat with? Or do I mean a fork in the road? Or do I mean a GitHub fork? I mean, those are all different versions of fork. They basically have the same sort of form, but that's not something that you necessarily, if you had a body, if you could see the world in addition to hearing the world, or if you could touch the world, these are things that we take for granted a lot of times in our concept learning, but they're there. And so again, it goes through embodiment and then social interactions, and so then all towards AI systems with human-like intelligence, the need for social environments. So we need there's social robotics, of course, as a field, but that's he's kind of arguing for this developmental robotics, which has been around for a while, but they need this sort of open-ended learning where they don't necessarily have a goal in mind, where they can just learn things, and they can interact, and they can do this with social robotics as well. So you'd have like social embodied robotics where they have a body, you have a program in a body, and they're interacting with each other, and they're engaging with the world and sharing information. That's the sort of the vision here. And so there are a lot of synergies here between developmental AI and cognitive science. And so psychologists have been discussing the idea that the brain might be motivated to explore novel situations or changing situations for many decades. And so, this is what I guess he views as fundamental, to look at these changing situations. And I think there might be a difference between like a changing world and maybe things that you haven't experienced yet. And so like when you haven't experienced something, you don't really have a context for it. It's just kind of like this blank void, but if the world is just changing and it's full of concepts that you already understand, then it's just simply a matter of transfer learning or something like that. But, this idea that you can go into a place and not know anything of what's going on, it's just this totally new context. That's maybe the place where, we really we don't know how to deal with that at all. So there are a lot of different synergies here. I'm going to put this link in the chat here. And so, and this is something I'll put in the Slack as well, because if people aren't in the meeting, you'll want to get a hold of this. I think I put a link to one of the things that, like one of the snippets from this, I don't know where it is, maybe down here further. This is Future Directions. It talks about cognitive bottlenecks. So there are a lot of things in this article. I don't want to go through the whole thing, but we'll come back to this at the end in the Slack review. Okay, so the next thing I want to talk about was, so this last week was Maine and Neuromatch, and then Friday there was nothing. And I did in the early morning, I did the tissue talks, so I went through those. And then, next week is, I think NeurIPS starts next week, or the week after, I can't remember. Looks like a lot of these workshops are the 13th. But I think the actual conference is next week. I'm not sure what the schedule is on that. But in any case, this is the list of workshops. So this is actually, we're at the NeurIPS website. So see if I can go back and get into the... Okay. So it starts, well, December 6th, Monday, it starts the tutorials. Tuesday through Friday is the main conference, and the workshops are next week after this coming week. And so this is the website for NeurIPS 2021. It's virtual only. I think it's like $100 I think if you're a student, it's $50. It's $100 for everyone else. I think Jesse and can't remember who else is volunteering at NeurIPS, but there are always opportunities for students to volunteer at these things. You work on some technical set of issues, you help them out, and you get free admission to the conference. They're going to have a lot of these talks on YouTube, which is usually the way they do it, so it's not like you're going to miss a lot of the keynotes. So it's always really good. This is the main conference in machine learning and deep learning and that sort of thing. So these are the workshops, and I think Charlotte Guo, who doesn't come to the meetings, but she's kind of like someone Jesse and I know, and she's sort of peripherally part of the lab. She's going to be doing some volunteering here as well. And they're going to be attending a workshop, I think, on meta-learning, which is this one here. 5th workshop on meta-learning. So, I mean, I kind of hope that we can go over that when that happens. So there are a lot of different workshops, algorithmic fairness, ImageNet, I mean, some very specific topics, AI for science, new frontiers in federated learning, meaning and context. We were talking about submitting something to this, but that never happened. This is pragmatic communication, machine learning and structural biology. So there are a lot of different workshops here. So this is something that if you're interested, there's going to be, we'll probably be talking about it in the Slack. There'll probably be stuff on YouTube about this, so I just wanted to bring this up. Okay, did we have anything we wanted to say about that before we move on, or?
I posted a couple of the workshops that look really, really interesting. For sure, there's going to be, we're going to have plenty to read.
Yeah, I know.
for the the holiday season.
Yeah. So I mean, and then I don't know. There's supposed to be this AI debate, which is where Gary Marcus takes on all comers, and he's like, and this is like going on a couple times, and they've done a couple of them where it's like, it's like at Neuromatch where they have the debates. The debates aren't really debates in the conventional sense. They're just kind of like, we agree on these things mostly, but maybe there's some things we disagree with. You know, and the whole thing has been like, you know, looking at like something like machine learning or deep learning, where you have like, you're just dumping data into an algorithm and you're getting an output. And that's one approach. And then there's the other approach, which is you need symbolic, you know, techniques that need to be there. And so then that's something that, you know, if you don't have that, you can't really understand, or the machine can't understand what it's doing. So those are the two approaches. And actually, that's how the MetaBrain stuff really got off the ground, was just kind of attending those debates and thinking about, like, you know, maybe not the middle ground, but like, you know, there's obviously a role for symbolic AI. There's this thing called Neurosymbolic AI, which is where you take the best parts of like a neural network and you put it into a symbolic system and you see what you can do. And so that symbolic system, of course, requires representations to exist. And then the idea was then, okay, we can build something with maybe different levels of representation. You have a neural net, which is very non-representational. You might have a symbolic system above it, which translates between the processing and the, you know, maybe like a topology here and some symbolic lexicon here. You know, you could have other ways of setting it up. And then you have this geometric relationship where, you know, you have like maybe there's a fold where you have the higher level representation surrounding the neural net. So it's like kind of going into different parts of the symbolic representation, accessing different parts of it that are sort of modularized. And, you know, like we know with the cortex is folded because, you know, there's space limitations in the brain, but there are consequences to that, and that you have these areas that are sort of in proximity. You have other areas of the brain that are, you know,
wiring constraints as well.
Right.
So I mean, back and forth between the wiring and the surface area.
Yeah. And so those are things, you know, that we could exploit that are not really typically talked about in AI. And that came up in Neuromatch, where they talked about, like, you know, how much of the brain do we need to model? And, you know, they have this debate going on, sort of a sub-debate where, you know, or considering kind of, you know, we could put everything in the model, but it wouldn't necessarily make sense. But there are some things that, you know, we can add that are robust. So people have experimented with computing with dendrites and things like that. So those are different, fundamentally different from like a neural network. And you know, but this is, again, I mean, an issue of like how do you abstract the brain, what the brain is doing and its structure and everything. So, yeah, I think that's a good, I mean, we constantly have this theme in the meetings of like talking about these sorts of things, and it's like, yeah.
But I wanted to add one thing, just because this comes up after conference time. You know, a lot of papers have been released early, you know, before. I mean, especially with this, you know, because there's a very rigorous review process. You know, the papers have been out. There's the GitHub stub for the software release, right, but there's no software release, right? And so we're gonna see this nice release of code over the next month, right? I think it would be a great service if someone made sure that code is actually runnable, and you can get what the project maintainers you know, hope you can from that code. In particular, what I wanted, and certainly I'm waiting for a couple project releases, is you know, what I would call, you know, the Breitenberg vehicle code projects. In other words, projects that are creating agents in a in a world that learn to walk and to do things. I think, you know, one, I think there's just some really exciting work in that, and two, I found previously that work can be difficult to get running.
Yes.
And for a variety of reasons, and like it would be a great service to have a server that's set up that's running those things that you can then interact with those particular projects, right?
Yeah.
So I don't have it here. It's my workstation upstairs, but a particular policy that looks you know, really attractive for learning, for you know, an agent that controls a quadruped. That, you know, I'd love to see. One of its dependencies is Mujoko, which is now open source. So you know, I paid for it years ago. You know, just getting that onto a system that is running, you know, should be doable now.
Yeah.
And yeah, seeing, you know, and even better, of course, and this is, you know, the kind of guiding principle of MOAB, the mother of all brain-computer interface benchmarks.
Yeah.
is the MOAB acronym. You know, it's even better when you can take 5 projects that all are making some contribution and get them running into the same world, you know, or get them running under the same environment and see, you know, looking at their behavior and things. That would be, you know, I think that would be a great, you know, not only it would be a nice like check on, you know, are these projects runnable?
Yeah.
But it would also be, you know, a great learning environment and potentially a great something you could build on for kind of next year for, you know, looking at performance across multiple agents.
Yeah. Yeah, it'd be nice if we could fork some of the better ones to see if we could get them to run, and or at least make a list of things we could fork. We could fork it in our orthogonal research lab GitHub, you know, not the ORL group, but the other one, and then we could work from the fork. Yeah.
So if I think it's Chris Holdgraf.
Oh yeah.
And is that right? So Berkeley person who's now runs this nonprofit or public benefit company that is running Jupyter hubs for nonprofits. So like their mission is to be is kind of like to be your DevOps, you know, and that yeah. And so I wonder if you know, one, they would do that for Earl, and I mean, I think you'd certainly qualify. And then two, yeah, it's like the only thing you need to add is for OpenAI Gym bracket all it includes Mujoko. Okay. So you also need to have this yeah, additional install past the the kind of Python project. But well, I think that would be great. I mean, you know, it's like that would make, you know, it really is. It is the vehicles. You know, I think there's a couple other. You know, it's not just OpenAI Gym. Like there's some other ones that you could install too.
Yeah.
But I'd like to, you know, like what's the minimum dependencies for like 80% of the agent projects or something like that.
Yeah. Well, hello, Jesse. How are you? Yeah. Okay. Stop by for a little bit. Oh, he's attending Machine Learning for Health NeurIPS Symposium. Okay, that's great, Jesse. This is one of the workshops. In fact, we are just talking about the workshops. So now Jesse's here. Why don't we go to the shared notes documents? I just wanted to go over some of these and just make you aware that they're here. So these are things. So these documents are things that I think we posted some links to this in the Slack. So just for, I think, Arthur's benefit, and maybe, I don't know if Morgan's familiar with this. The idea here is that we have these docs that are just kind of doc files, and if you're attending this conference, this is the main conference, you can put in all sorts of notes here. So these are mostly screenshots of different talks. So, and then I think for main, I mainly did this. There were talks by different keynote speakers. So this was a talk on next generation recurrent neural network models for cognitive neuroscience, just having some screenshots in here. And this isn't necessarily efficient, but it gives you like a place to pause, and maybe something you found interesting, you can leave a note here. And then anyone can do this, so we can compare notes at the end. Let's see, I had some notes in here on some things. There was one really good talk. This was Josh Vogelstein, Lifelong Learning and Beyond. So he talked about sort of this idea of lifelong learning. So this is like long-term learning in an agent, and kind of going through some of the key references. So this is a paper, A Theory of the Learnable. I didn't get the full citation, but these are some classic papers here where they talk about, sort of learning. And of course there's informal and formal learning, and it kind of makes that distinction. And then they implement this in a reinforcement learning agent. So they have, they kind of walk through a lot of this from that perspective. I thought there were some interesting things here. And again, if you look through these slides and you find something interesting, you put a note by it. I've just, put in a number of different slides that I thought might be, takeoff points for conversation. So, there's some technical detail here, but, and then 30 years later, there's some things on catastrophic forgetting. So these are, of course, continual task learning. We've actually had some conversation. Of course, we have this paper on continual embodied learning, but there's, I think, a lot of interesting stuff going on with continual learning more generally, which is this idea that you can, you have learning that just kind of proceeds over a long term. You don't have it on specific tasks or specific trials, that it's a cumulative thing. And so that's interesting. Then there's another talk on, let me go down here. This other speaker on a lot of stuff here. Well, this was Bing Brunton, who is also at Neuromatch. She did some things on turbulent plumes, which are when insects or some other invertebrate tracks a plume either in water or in the air, and they're usually associated with smells or some sort of odorant. Sometimes it's a liquid, but in this case it was an odorant plume that this insect was tracking, and they were looking at this with deep reinforcement learning. So this is kind of a mix of ethology and reinforcement learning. It's really interesting talk.
As well as physics-inspired neural networks.
Yeah, definitely. So this is an odor plume. You have this place where that follows the wind, and this the insect is navigating along the plume, so it's got this kind of weird path that it's taking, but it's basically trying to track the plume as it's wafting through the air. And so that's the challenge of this. And of course, when you have turbulence, when you have like wind, it makes the plume more sort of diffuse, and so the insect has to really kind of work to follow this to the source. And so the source might be some tasty rotting fruit, but they want to find out where that is. And so this is, it involves a lot of sort of sensory integration. You have to see things and smell things and track them, and then plan, and then, plan to land in the place where this odor is coming from, and so forth. So there's a lot of that. Let's see, there was this other talk on, talked a little bit, yeah, neural dynamics, talked a little bit about emergent behaviors and embodiment. This is on Surya Ganguly, who talked about foundational system science challenges. So this is where he actually worked with McClelland, who was one of the pioneers of some of the backprop stuff and the PDP stuff with neural networks. And they actually buried deep in our developmental Bradenburg vehicles paper. There is some review on some of this work that McClelland did with some collaborators on sort of these developmental neural networks. And at the time, they're basically neural networks that have this developmental sort of model for processing. And so in this case, it's about acquisition of information at different stages. And so that work is like, you know, probably 30 years old now, but it's interesting work. And he kind of followed up on that because he talked about development in this talk here that he did. You know, he kind of went into some of the systems like the Retina and reverse engineering the Retina. And he had some interesting things to say about cell types and why the Retina has the diversity of cell types it has. So this is an interesting question from a developmental perspective, which he didn't address in this talk. But, you know, why is it that the Retina has multiple cell types? Or why does the liver have multiple cell types? You know, is this like purely a functional thing? Or is that just kind of like how the Retina works? I mean, you know, we don't really know why the Retina has a lot of cell types. I mean, he kind of addresses it here. But I thought this answer that he gave was more sort of because function. And of course, that's not, you know, that's not necessarily an answer. If you observe a retina, you know, in an eye today, and it has this diversity of cell types and it works, and, you know, you can see it in development. You can watch how these cells differentiate. But then you have to add this evolutionary aspect, which is where you say, okay, how did these cells first come into association in a retina? How do you end up with these cell types? Is it that you had some precursor cell type that then started to differentiate into different niches in the retina? So, you know, there might be a need for maybe like a cell to be tuned to one set of light frequencies versus another, or there might be like, you know, some, you know, a specialized role that some cell plays. You know, so these are things that and he didn't talk about this because he's not a developmental biologist, but I think it's interesting from like an artificial systems perspective. You know, can you model these different cell types in the retina as they're emerging? Not necessarily given like a goal of looking like a retina, but just kind of like an open-ended evolution where if you just give it certain conditions, what kind of cell types do you get in the retina, in an artificial retina under certain conditions?
And of course, many developmental biologists see development as a way to probe evolution, right?
Right.
Yeah. So they're certainly, you know, very intertwined. I'm curious when, yeah. I mean, do we have any good examples of neural networks? You know, I mean, I guess I'm thinking a little bit of sort of, is it Richard Watson? Or maybe maybe this is Jeff Clune, but the development of modularity in neural networks.
Oh yeah, I think Jeff Clune does work on that.
Sorry. Yeah.
Yeah. Oh yeah, I think so. I think he's worked on that. Yeah, there is this sort of.
Yeah, I mean, you know, so yeah, thinking of Jeff Clune and I think some work he did with Murray, the ResiBots guy.
Okay.
They looked at modularity, but you know, do we ever allow like a neural network to basically like duplicate itself and then and then allow that, that new duplicate to develop separately,
right,
in that kind of, modularity, but then separate, separate development, right? Because I mean, I'm just trying to imagine, building a brain, right?
yeah,
and like build, having a simpler brain, and then having, some sort of duplication, and then allowing one of those brains to get more complex and the other stays, the cerebellum or something.
Yeah. That's kind of what we've been trying to do with our developmental Brainberg vehicles, is you have like, this encoding that produces cells that come out, and you could have like, different modules that, that are defined. You just say that there maybe one module at one point in development, and then there's a split where at some point in development there's a shift where they just become disconnected, and then they have their own sort of inputs, or they become, conversely, you could have things that are disconnected then become connected, and then that's something that maybe activity dependence will so like you say, okay, nominally these networks are separate, then they become connected, but if it's in an environment where it's not getting any information about certain things, like you only get information on one side versus another, then they remain separate. So you can do things like that. And there are people who, I don't think there's anyone who's really ever addressed that question. I know that there are people working at the intersection of like artificial life and neural networks, but there aren't that many people, so I don't think people have ever looked at that. Yeah.
But
oh, go ahead.
Oh no, sorry. That's just my daughter.
Oh, okay. So this is just more on this, and there is some more stuff here on development. I thought that was really interesting. If I can find it here, it's a little further down. And so, yeah, there's this fourth question where, how and why do many aspects of infant semantic development also arise in deep neural networks? And so he did this really interesting stuff with, with looking at like the emergence of things in the retina and development. It was kind of an interesting take on it. This is another thing that talks about 70% of all cell types in the primate retina are comprised of these different types of cells. And this is an interesting theme that came up in Neuromatch too. And they're talking about like how you don't have 100% of cells doing something necessarily in its structure. Sometimes you have different cell types, and maybe some of, there's a proportion of cells that are doing something and other cells that aren't doing something. And so in this case, you have different cell types in the retina that are doing things, and there's a proportion of one cell type to another. But then also there was a paper, I don't even know, maybe I'll switch to it now. So there's this classic paper, this one, that says, what is the other 85% of V1 doing? And so V1 is this like first layer of visual cortex in primates that is a subject of intense study. It was a subject of the Hough and weasel work where they were looking at opponent stripes. So basically in V1 you get this detection of stripes or patterns, rudimentary patterns like that in the visual scene. And so this is dependent on cells doing this sort of pattern recognition, this pattern formation, and this sort of processing. But the problem, of course, is that upon sort of further study, people found that maybe only 15% of cells are doing something related to this sort of pattern formation. So what are the other 85% of V1 cells doing? And this is the question that they ask in this paper. This article will pose the following challenge: that despite 4 decades of research characterizing the response properties of V1 neurons, we still do not have a decent picture of how V1 really operates. So this whole region V1, we know that it does this thing that Hough and weasel have shown pretty convincingly that it does, but not all cells are doing this. We know that some of the cells are doing this, and it's the most salient thing that it's doing, but we don't really know how it really operates. So how a population of its neurons represents natural scenes under realistic viewing conditions. So we know that it's doing this one thing, which is like finding stripes, but there are probably other things it's doing we don't really know. So, yeah. Go ahead.
Well, I mean, it just, thinking about GP Wright and the kind of, theoretical DNA research, right? Like, are those neurons doing it?
Right.
Are those other 85% necessarily doing something when we've just made the argument that this is an evolutionary structure, right? And it's definitely trying to be efficient, but its main selecting force is functionality, right?
Yeah.
So if it has to have this extra baggage, but it creates an organism that can see and can respond to its environment in a timely fashion, right? Then, right, like mission accomplished.
Yeah.
And maybe it's it takes like the other 85% is like metabolic.
Right. Yes, thank you for bringing that up, because that's exactly what I was thinking.
Okay.
And I mean,
this is the junk DNA hypothesis, or the things that come out from what's the big project, Encode?
Yes, Encode. Yes.
Yeah, anyway, just like I wonder what parallels perhaps exist there.
Well, one thing that, like, and I think about cellular reprogramming too, because that what basically happens is you get a proportion of cells to reprogram. You try to you introduce the factors to all the cells in your plate, and then only a portion of those survive and actually reprogram. Now, in this case here, it's because there's differences in uptake and all this, but really, you think about, like, in a functional context, you want to have redundancy. You don't want to have like just one cell to one thing, right?
Yeah, I mean that's a great point, right? And that unless you do start knocking out regions, right? You don't realize that there is this redundancy, and in fact, you can remove half a child's brain and have function return. With a hemispherectomy, this is a much broader, much larger than V1, but that that there is a lot of redundancy in there, and certainly when what is it, the Innovative Genetics Institute is using a laser to take out all the cells that don't program correctly. Right. The biological systems don't have that laser.
Yeah.
And I have to come up with other mechanisms for handling, the same problem.
Yeah. Okay, Jesse had some updates here. So let's see. Congrats to everyone on the Neuromatch stuff. I think the Flash Talk Discords were not really well advertised. Yeah, there were a lot of them in the Discord, so but they got more responses in Twitter than Discord. Was still a good event. I think they were just experimenting with the Discord server, but yeah, I mean, I think it was a good place if you want to chat, but it's not, it's very overwhelming when you have all this stuff here. Did anyone get to see the Day 2 panel on study learning versus computation? I had to miss that one, but we'll check it out. Yeah, that was the one, the last one, right, Jesse, with Conrad Cording in it. I think I had a couple notes on that, but yeah, I saw that. Okay, you're going to probably be out next week a little bit. That's fine. Another lab admin news, I'm going to revise and update some of the onboarding materials, including ways to contribute. That's very good. Thank you. I know we're always trying to keep on the cutting edge of that. Avery will be turning to the U.S. soon. Collaboration with Jenny is still ongoing, but I know she's also dealing with grad school apps. Everyone's always, yeah, it's always a tough thing. Anusha was going to come by today to the meeting, but I think she had another thing come up. Okay, that's fine. Yeah, whenever she can make it, and if she wants to talk offline, that would be great. very curious at what Timnit Gebru does at her new DARE research group. Yeah, she's Timnit Gebru, of course, is the person from AI ethics, and she was fired from Google, and she's now got a new research group that's starting up. And so, yeah, that's great. Yeah, it'd be interesting to see what happens there. I attended the last meeting of the year of the AI ethics research group at MIT, and it was good. Write up about that soon. Yes, please, if you could actually share it on the Medium, that would be great. Very interesting to look at people in the trenches trying to deal with current points of interface of what is tenable. Yeah.
Well, just gonna say, you know, she was fired from Google for saying that Google had bias problem.
Well, yeah, I know that was.
I mean, sorry, I know you know that, but
yeah,
just so that it's stated.
Yeah. So yeah, actually, why don't I go to the next? Yeah, so I think that was main. Oh, okay, I shut my screen share off. Let me go back. Then yeah, we have another document for Neuromatch. So this one was Neuromatch. This has actually an index in it. So thank you for doing this, Jesse. I know you were working on this index, and we had some really interesting things here. I don't think I put the notes on the last panel on here, but they're at the bottom of this. But Danny Bassett, of course, gave an excellent keynote on some of the stuff on actually modeling cognitive effort as a network and a network perspective on that. She brought up the Hurst exponent, which I haven't heard about in a while, but it's a very popular method in physiology to look at different components of a time series and dynamical systems. So she actually had an approach to network controllability. And I know Albert Barbasi, who is a very famous network scientist, has done a lot of work on network controllability. And it's a different perspective than what Danny shows here. So this was a nice talk. Talks about state changes in dynamical systems, which is really good. State transitions in networks. So, you know, this is all in a network. And so I think there was a lot of good stuff here to go over. And of course, talking about minimizing energy costs. This is great. Okay, so then there's this other part here on, that's not the one I wanted. Go down a little bit more. Yeah, there's some good stuff on this. This was, I don't know if this was the talk here, but this is about insect navigation and yeah, and there's this inside an all-natural deep recurrent neural network. This is, I think, this is based on the Fly-EM data. So there's this new connectome. It's the Drosophila connectome, which is much bigger than the C. elegans connectome. And Genelia, which is the big lab that the Howard Hughes Medical Institute runs, they've done a lot of this work. So they have these electromicrograph images, which are like microscope images of the cells, and then from there you can infer the connectome. You have to do some analysis. You know, there are ways that they do this, and then they determine the connections between the cells in each slice, and then they can build these models, which are these huge 3D models of the brain, and so they can then, you know, determine where the cells are, the connections, and then build these really beautiful models of, you know, cells that are all connected. And then it's up to the person, you know, someone who wants to analyze it, what that means. So they can build the model of the brain, but then, you know, what is it? And of course, this is an insect brain, so you have mushroom bodies and, you know, different parts of the brain that are different than what you might find in a mammalian brain. So this is a nice, I think, you know, a nice bigger connectome than C. elegans, and something you can definitely compare with neural networks. And here they have one, fully trained deep recurrent neural network for flexible navigation. They have it kind of laid out in this talk. So there's that. But then I wanted to get to this other thing. Where was it? Down a bit. Those are models and data, the short talks. This is an example of the complete connectome of the insect brain. So this is a model here, what I call it in the Slack, gumballs and cotton candy. It was like, because you have the gumball on top and the cotton candy on the bottom. It's just the way they visualize it, but it's, these are neurons and these are nerve connections down the, so this is the disembodied brain of a fruit fly. And then you have these different motifs that they've identified. So again, you're looking at a network, you're looking at different little components of the network, and they've done a lot of work on this, so it's really impressive stuff. Let me see if I can get down to the bottom here.
And just to add that this does kind of exist for human brain, or certainly, a big brain is I think the closest equivalent to this work that you can find.
Yeah.
And is a massive data set that a lot of the Genelia and Allen Institute people they've had kind of workshops and sort of hackathons of the between these groups. So again, like I think they are sort of building on some of the blue brain work.
Yeah.
And again, like a lot of the Genelia people showed up for Neurostorm, which was covering how to tackle the human brain. There's only one human brain that's been looked at with this kind of resolution.
Right.
So it doesn't lend itself to that kind of. I assume that they've got more than one Drosophila brain with that cryo.
Yeah, I think they have to take many brains and then get them to kind of, get the images and then kind of figure out, like, there's a lot of automation in this too.
Yeah.
So
I mean that's what Neurostorm was all focused on, was like how to do this at scale when we've got the big brain data size itself was bigger than all the MRI publicly available data sets combined.
Yeah.
Right. Including UK Biobank.
I have to say this is vastly different than some of the stories I've heard about the C. elegans work, that like a lot of the connectome work and a lot of the cell work that was done was actually done by drawing pictures on paper and then like taking those drawings and like matching them up, and so it was definitely very different from what they're doing now. It was not high throughput.
Right. I mean, well, isn't that kind of like Image J kind of was popular, but people were still like drawing on the images, but just digitally, right?
I don't even think they had Image J then, because this was like in the 70s. Some of the stuff, like John White and some of the other things that they were doing then. But this is like, you're talking about going from that to like these huge scanners and, electron microscopes are doing it all by computer, and you're kind of finding the annotations, and so it's a different kind of system. But I mean, still, you're getting, you're still having trouble finding the biological insights sometimes. That's the lesson, I think, is that, it's not automatic, just technology.
Yeah. I mean, they very much were concerned with, hey, we've got the same patch of cortex at like micron level, but we've got it with like 10 different stains, right? How do we make sure that those all end up on the same neuron? Yeah. Or how do we co-register those?
Yeah.
That was all they were. Forget about biological function.
Yeah. So I had one more document here, the tissue talks, but this, I'm not going to go through this too much. I just wanted to make people aware. So again, this is another note document. This is the tissue talk from this past week, which one thing I found about this that was really ingenious was that they did this study where they didn't actually reprogram the cell in terms of like the stemness pathway, but they reprogrammed and I think I understand this correctly, the cell cycle. So they were able to take so cardiomyocytes, which are cells in the heart that are differentiated, they don't regenerate. So, you can't really, you can put stem cells in the heart if you, say, have a myocardial infarction, and the cells basically just, when they undergo this cell death, they just become scar tissue. So you can put stem cells in there and hope that they differentiate into heart muscle. Or you can reprogram cells that are there, turn the cell division or the cell cycle back on, so that gives you more cell divisions, and then hope that they reproduce themselves in context. And so in this case, what they did was they delivered factors that would maybe turn the cell cycle back on, and they were able to get 15 to 20% of the cells undergoing stable cell division, meaning it's not cancerous, it doesn't go off the rails like a cancer cell. And they were able to get increased cardiac output, which means that it was actually functional and doing something beneficial. So, and again, you see this 15 to 20% figure. Again, like I said, these things are probabilistic. They're not like 100% or 0. And so that's an important thing to remember about a lot of when we talk about like cells in the brain or anywhere else, or anything that's, biological. There's always a probability to it. It's not a all or nothing thing. Occasionally it can be, but that's, that's not really the rule. It's the exception. So yeah, they're able to get cardiac fibroblasts too. They can reprogram into cardiomyocytes. There are fibroblasts in the heart that exist, so you could actually reprogram those as well. And so there are this was a really cool talk. They talked a lot about sort of stress on the heart and how they can, repair tissues and things like that. So it's not organoids, but it's close. There are a couple of papers in here too. I've taken the abstracts and put them in. So these are...
Did you get this from the speaker?
No, I got it from the YouTube channel. There's a YouTube channel here.
Oh, okay.
So
and
this
they added that to the YouTube. Okay, awesome.
It's, I think, Columbia BME, so it's the Biomedical Engineering Department. But
yeah. I mean, this and Tarasky and I think the Tarasky would be the West Coast best version.
Yeah, so this is available as well. And I have these all in the Slack. I don't know if you need any of these documents. I can make, I can repost the link, but I think that one is in Brain Organoids, and then the other ones are in the, I think one's in the Neuromatch channel, and, so we can definitely, if you want to look at these or add to them, I can send you the link. Yeah.
Transmedtech is the Montreal tissue engineering, or definitely has a big focus on tissue engineering.
Okay.
It doesn't have to be their only thing, but I've been trying to catch these live, but I wonder. I mean, I should check YouTube and see, especially if they're adding the presentations.
Yeah. Sometimes it takes them a while to get it up, but
Sure.
Wanted to actually, I had a couple other papers in this 15% of cells, developmental stimulation and critical periods folder. So there was another paper on, we talked about bistability in the switching presentation, and this is a paper that I did a little bit of digging in, watching some of these things this week. This one is bistability in feedback circuits as a byproduct of evolution of evolvability. So what does that mean? So noisy bistable dynamics and gene regulation can underlie stochastic switching. So this is what we were talking about in the talk, that there are examples of this in gene regulation where you have switches between different promoter regions and different gene products. And, there are things that they're noisy in the sense that they're stochastic. So in other words, you don't have like a deterministic switch. It's like one set of environmental conditions might result in the switch going off or not. It's a probabilistic thing again, where it could be like 20% of the time the stimulus will trigger this thing to happen, the switch to go off, but 80% of the time it doesn't. And then if the noise is stronger, if the signal's stronger, then it might go off more often. So this is what they mean by stochastic switching. And so you see this in fluctuating environments, where you have an environment that's fluctuating constantly. You have like, it's really hot or it's really dry versus not. And these environmental conditions or there's a lot of stress versus not. This is how this stochastic switching becomes useful, because, if you're experiencing stress, you might want to have this switch go off. But if you're not experiencing stress, maybe you don't want the switch to go off. So and without putting it into a specific context, that's how it works. But we don't know if this fluctuating selection can result in bistable dynamics. So we talked about the bistable dynamics in the switching presentation as well, that you have this ability to go between two states. So, usually a bistable system is poised in between the states, and then given like some stimulus can go to one state or another state. And so it's bistable. It's stable in two states, and then, it depends on what's going on in the environment. So they use a stochastic model of a simple feedback network. They apply fluctuating selection on gene expression and run evolutionary simulations. They find that independent of the specific nature of this environmental fitness relationship, the main outcome of fluctuating selection is the evolution of increased evolvability in the network. So this is kind of what we're talking about with redundancy. So there's this interesting relationship between redundancy, evolvability, and robustness. And there have been books on this. We can talk about it if people want to explore this in more detail. But the idea is that redundancy allows you to do, like, to have several different mechanisms for the same thing. So if one mechanism gets, killed off, or if it doesn't work for some reason, the other mechanism can take its place. And so you always have that ability. It's just run by different mechanisms. This allows you to be robust. So when you have a redundant system, it tends to be robust to environmental perturbation. So if you have, like, say, a drought where, your normal pathway doesn't work, you have another pathway ready to go that can substitute. We talked about this in the brain, where if you, obliterate parts of cortex, other parts of cortex can take over and substitute. Or there are other, things in the brain where other regions can substitute. It's not like unlimited, but there's this definite relation. You have this robustness that occurs from redundancy. And then this feeds to evolvability, in that if you think about this evolutionarily, you have this ability to evolve. So if you have this redundancy, maybe in mutational redundancy or functional redundancy, that means that you can evolve more potential. Your evolutionary space increases because you're not locked into any one pathway. So, without kind of going into a specific example, if you have a very tightly regulated system that's like hand-coded for one thing, if you're very specialized, you can't really evolve anywhere because you don't have the background, or you don't have the redundancy to sort of evolve in different ways. You don't have those multiple reservoirs of variation that you can draw on if you, say, need to escape. If you're a very highly specialized insect, for example, and you need to escape your environment because it's going away, it's drying up, or it's heating up, and your niche is gone. So you move to a different niche because you have this redundant reservoir of mutations and other mechanisms that allow you to, allow at least part of your population to survive. And so that's what evolvability is. And, we've talked about this, of course, not just with mutations, but at other levels of complexity, like in terms of neurons or in terms of functionality. So this is their results provide the first direct evidence that bistability in stochastic switching in a gene regulatory network can emerge as a mechanism to cope with fluctuating environments. So that's this paper is. And it gets pretty deeply into some of this jargon on like evolvability, but the idea is that, this stuff we've talked about with switching, the stuff we've talked about with a lot of the redundancy and other things, they're all related together. So this might be a theme that we could pick up to think more deeply about some of these artificial systems and how to improve them. This is another thing. This is a classic paper on critical period stimulus input and emotional reactivity. This is something that came out of one of the I think it was the Maine conference, and Alison Gopnik was talking, and I think there was another speaker who talked about development as well. And they weren't really like hardcore AI people. Alison Gopnik is a developmental psychologist, but so this is on infantile stimulation. So one of the insights that Alison Gopnik made was that behavior or these mechanisms are different in children or in development than they are in the adult. And so we've talked about this in the lab a lot, but there's this, they used attention as an example, and I think I actually have the slide in here somewhere. It's down a little bit further, where there's this, see if I can find it. This is her talk. There's this attentional, okay, here it is. This is attention, or she calls it consciousness. There are a lot of parallels between consciousness and attention, but this is, I guess, consciousness, but it's also kind of attention. There is this idea of the attentional spotlight, which is where if you're looking at things, you focus on something to pay attention to it. So if I focus on this desktop computer, I'm focused on it, and I see it, and I'm aware of it. But then there's stuff in the periphery of my vision that you don't necessarily see or are aware of. That it might be happening 5 feet away from you, and you don't necessarily notice it because it's in your peripheral view. So this is what they mean by the spotlight of attention or of consciousness. You focus in on a certain point. But as it turns out, that's only what happens in the adult. In infants, you actually have something called a lantern. And this is because infants are taking in so much more information that they just, infants kind of dart their eyes around and look around and explore. And they have this model of attention or consciousness that's this lantern. So it's like this big circle. And so it's very different from the spotlight. And so her point was that consciousness narrows as a function of age, that you get this spotlight that forms over, life history as you move out of development and into adulthood, you get this change in how the brain functions. So this is where this paper comes in, where you have stimulation of infants. I think in this case, I think they were talking about rats and mice, but also humans. So this ties into the critical period stuff and stimulus inputs. And, I'm thinking about this also in terms of Brainberg vehicles, because there was some stuff on vehicles, actually, I think in one of the Neuromatch talks. And it was interesting because the vehicles were kind of mentioned in the first couple slides of one of these talks. Here it is, this one here. This is the all-natural deep recurrent neural network architecture for flexible navigation. This is where they got into the connectomes for the Drosophila, but then they started with vehicles. They started with these very simple vehicles here, and then getting into how this vehicle, this black box of the vehicle nervous system, which can generate, so the vehicle, it orients itself to basic stimuli. It also performs these behaviors that look like things like love or shyness, which are just basically like, they call philic and phobic behaviors. So it's like light philic or light phobic. And so, it's either shying away from the light or it's attracted to the light. And that's the idea. The Brainberg vehicles do a lot of those type of behaviors. If you go to Brainberg's book in 1984, he talks about these different behaviors that he's classifying these vehicles as doing. So it's like love, hate, shyness. They're not like bona fide behaviors in the neural substrate, but they look like those sorts of things. So what this person was saying is that you can take this black box of connections and you can, substitute a connectome, and you can, so he's kind of making this point. He didn't return to the vehicles metaphor, but I just thought I'd bring that up because I think it's, I think we should think more about that connection.
Certainly, neurorobotics.net is absolutely doing, trying to do that.
Yeah.
The fact that they're using ROS, robot operating system or robotic operating system, is kind of immaterial. Like the main thing is that they've got a neural net that's the agent.
Yeah.
Right. In the simulation, and they're definitely trying to. They're just trying to do a 3D version of Bradenburg.
Yeah.
So, I just I came in at the end of that thing. Was it Vivek's talk? And what were you showing? I kind of missed. I just saw the Bradenburg vehicle stuff. What was the context of all that?
Well, this was just the this is the Bradenburg vehicle stuff.
Denver.
Yeah. So this was Vivek mentioned Bradenburg vehicles. He did some talk about like the Drosophila connectome. So this is the fruit fly, and you have this thousands, on the order of thousands of neurons, this connectome that they've been able to map out. And so he's looking at vehicles, using it as sort of a metaphor for what they're going to do with their connectome. He says you can replace this black box inside the vehicle with a connectome. He didn't do anything more with it. But I was thinking, like we talk about developmental Bradenburg vehicles and how they have this, what it might end up being able to do. If we have this developmental component, if we play around with the internal network here, and this is just very simple, this isn't like this thousands of cell connectome, but just what can you do with a very simple nervous system in developing it. So.
And Morgan was something about the 3D vehicles or something?
Well, I was just saying that neurorobotics.net is definitely trying to make an active software project of vehicles with a it's not a connectome, but it's actually just a neural net, right?
Right.
But just back to the Denenberg that you were showing, and how that relates to the Gopnik. Seems really interesting.
Yeah.
In that, I'm just looking through the abstract here of the, where you do see. I mean, Karmeloff Smith talks all the time about these U functions, but that critical periods could be seen as a narrowing of attention. Or I think he's trying to reframe what a critical period actually means. Is that right?
I think so, but because we have all this stuff on critical periods too, where we're looking at, like, some people are looking at like the molecular pathways for critical periods that there's this window that opens up where there are different things that are expressed, and then it closes off, so you can where there's like synaptogenesis that occurs at a certain time. But then critical period also has a sort of behavioral, because this was back in like 1964, so this is like more of a behavioral thing where you're looking at like general learning or general changes in behavior that are happening at certain times. So. And it's a little bit different definition, and I think people do play around with the definition a bit from what I've seen from different papers in this area, like just what they call the critical period.
And this is what we don't see, right? Is that like there's too much work focused on learning a task and optimizing a metric, right? And to make the point. I can't remember the researcher, but certainly, the people that are collecting data of infants over periods of years, right? That they don't just learn the task, but then they're still alive and they're still taking in new input, and what in sort of the continual learning framework, you know, what happens to that neural network? You know, you've learned the task, and but you keep going.
Yeah.
And you know, and you kind of, I mean, this is certainly Karmeloff-Smith's point, is that like you want to see those U-shaped, I mean, not inverted U, but you want to see those U-shaped behavioral growth patterns, because that's indicative of, you know, whatever you want to call it, a critical period, a bifurcation, or, you know, some sort of new dynamics.
Yeah.
In the system, right? That the system has learned something, you know, to use the typical example, you know, of past tense verbs. They've learned how to do something. It works most of the time. then it becomes kind of overused, and it's failing in a number of edge cases, or, you know, irregulars, right?
Yeah.
And then boom, you know, something else happens, and performance becomes, you know, almost perfect, because again, it's not that it's just generalized, it's that something else is going on, right?
Yeah.
And just
yeah. Oh, I think in this U function, you know, this is like the classic arousal U function, where you have this optimal point where, you know, you have you know, too little stimulation, too much stimulation, and then like this. But it's interesting to see how these things would change over development, in terms of just models that we use in adulthood. You know, how do those things change in not only in development, but in critical periods where you're acquiring a lot of environmental information. So like, yeah, I think this is definitely like where they're kind of going, you know, like you just don't have experience with, you know, noxious things in the environment, or you don't have experience with the environment, so your performance is going to be different. And so, yeah.
Right. Okay. Yeah.
Yeah.
But do you see that in a kind of a continual learning framework?
Oh, I don't know. I don't think so. You know,
you know,
well, I think, yeah, it's definitely something that's part of that continual learning framework, especially in terms of like, you know, if you're taking a developmental stance on it and saying we want to have an agent that sort of not just learns things, but develops as it learns. So it's learning through developing. So it's like just starting from maybe some basic primitives, and then it's just kind of, you're letting it go. And, you know, you don't want to just train it on a static neural network. You want to have...
Yeah, no, I mean, I'm just wondering, like, do continual learning people talk about, you know, going from a network, going from a, I don't know, perceptron to a network that includes attention? You know what I mean?
Oh, no.
Yeah, it like yeah, because I mean, I guess that's sort of Gottnick's point.
Yeah.
Is that you've gone from this, you know, I'm taking in everything, or, you know, I'm taking in everything and performing unsupervised learning, and then at some point you've got this narrowing, and there's this attentional filter, and
right.
I don't know how much, you know, work there is in developmental AI for computationally, you know, how much work exists for that. I mean,
I don't think there is. Yeah, it definitely. Okay. Yeah, I don't think that's a perspective people have really thought about too much, because, yeah, I don't. Yeah, but she was coming at it from like a developmental perspective as a psychologist, and she's even said, I don't really know a lot about like the cutting edge of this area, which is, you know, but I mean, I don't think that like there's a reason why they invite these people to the AI conference.
and it gets at, like, in a sense, you won't reach that,
right?
Because you're never making a, you're never making kind of your computational world big enough for, or a framework that can allow for that kind of stuff to even happen. And yeah, it's like if you're seeing it as matrix theory, then like you won't.
Yeah.
Sorry.
Yeah. So I think this was what Jesse was talking about here with the Vivek and then Denenberg. Yeah, okay, so we cleared that up. All right, so one more thing I wanted to do before, and then I wanted to leave some time at the very end for, we were going to talk about NeuroHack or some of those things. So, but I want to go through one more thing before we get to that. So this is another thing, and I thought Jesse would be interested in this. Okay, I have two things actually. This is this paper I put in the Slack, charting the future of neuroscience. So this is a paper that was charting the structure of neuroscience. This is one of the panelists at Neuromatch, and this is one of his papers where what are neuroscientists actually interested in? To find out, I studied their itineraries at the annual meeting of the Society for Neuroscience. So this is the structural graph that he built. It's just a bunch of points sort of clustered in space. So this is the map. You have all these different categories. This is SFN, so it doesn't have everything. It doesn't have like economics on it, unfortunately. I know that Daniela liked this, but I don't think they have neuroeconomics on here. But this is like a lot of the medical research, a lot of the Alzheimer's disease, Parkinson's disease, glial mechanisms. And then you have like stress in the brain over here, pain. So you can see it's very SFN oriented. There isn't a lot of computational stuff. Olfaction and taste, homeostasis, brain blood flow, or vision, it's clustered in here. You have learning and memory, hippocampal studies, animal cognition and behavior. So you have all these things over here. It's kind of like the cognitive area. Then you have like the biomedical area, physiological area, brain-machine interfaces down here. That goes along with like motor control. Then you have vision, you have visual disorders, you have drug abuse and addiction. So this gives you like a landscape or like a mapping of the field. And this, I think, Jesse, you might be interested in looking at this a little bit more, just in terms of how these things look, how far apart they are, and what kind of clusters together. This is the paper here, so I can post this. I think I did post the paper, so you have that. But that's what that was about. And then the other thing I wanted to go over was this. This is something I found. So there was that whatever happened to cognitive science paper. This is a paper by Peter Osaro, and as far as I know, this is something that isn't published anywhere. It's on the web, but it's I just ran across it, and it's called Whatever Happened to Cybernetics. And this is a nice overview of cybernetics, sort of like starting from the 40s. And then there's this narrow view of what cybernetics is. And then there's this broad view that he gets into down here. So the narrow view is that that's the mechanism of negative feedback control. It's metaphysically central to the order and organization of the universe. And then this broad view is, the broad view starts from taking the rhetoric of cybernetics seriously, and then this kind of goes into some of the stuff that's going on currently. This guy, I think he's some sort of person from the humanities, so it's a bit, oriented towards like how people have used it in the humanities as well as in science. So, this might be something, again, that there's also an internal view and an external view, I believe. So, is cybernetics dead? This kind of looks like something Rob Stone would write. So, yeah, it's definitely something you might want to look at. But, yeah, and then speaking of Rob Stone, I've been working on a revision of our Every Good Regulator theorem paper. And Rob doesn't really attend meetings, but I'm still working with him on this stuff. So I sent a draft to Jesse. He's looking that over. And maybe when we're ready to share this paper, we'll go over the new version of it. This is a paper that we've been working on for a couple years, where we're looking at, like, this ever good regulator theorem in cybernetics. It goes back to about 50 years. And it's this idea that there's this regulation, that they kind of give the conditions for good regulation, and then we kind of build on that. And so it's kind of an interesting paper. So I know that Morgan brought up BrainHack or NeuroHack in the chat. So you want to talk a little bit about that?
Sure. So yeah, these are two different events. One, BrainHack is ongoing. Started November 28th, I believe. Globally in Australasia, you know, since they are where the dateline is, and so I think Sydney or Melbourne is the center there. They might be done at this. I mean, I think they're done, but you know, BrainHack is a set. I mean, it's BrainHack Global, and it's running from November 28th to I think mid December. But this is multiple local chapters, right, or multiple local events, and so depending where you are, you know, check, and there might already be a group that has plans and is setting up something. Montreal just changed their event to be online only.
Yeah. So people, is this something that people can join in, or if they're not, do they have to be registered, or how's that work?
Say that again. Oh yeah. I mean, so it's register. I mean, it's free, but register. You know, so they want to know who's there, and as I have, maybe this is in NerdTechX. Somebody asked, like, why do we have to register if it's free? And reporting, you know, that this is how academics get funding, is, you know, hey, we did this event and 300 people registered, or, you know, Maine like had 2,000 registrations, you know, and that is super valuable for them in terms of getting support for next year's Maine, right? So, you know, that they could say that there's like 2,000 registrations from 70 countries, like that as well, you know. And this is exactly why in neuroimaging, SPM and EEG lab are free and are GPL, but require you to go through a registration screen, you know, before you get the download link. And some of that is that they really want people to register. So, you know, you know, as it comes around to grant renewal time, they're like, yeah, please tell us you're using our tools. Yeah. So check out Montreal, because Montreal just switched to being online only.
Okay.
I think a lot of groups should consider that. You know, like everybody seems to have gone back to business as usual, and you know, we could be heading into an even worse wave, yeah. Which is scary. And anyway, it's the reason I dropped it into the chat was that I think they were also saying, like, hey, if you don't have a local chapter, like, please sign up and pick a project and help out. This is definitely, like, you know, more than anything, this is a hackathon. You know, this is like, find a project and make some contributions to it.
Right.
You know, and NeuroHack registration has ended, but it's going to be really interesting to see what NeuroHack. Sorry, let me. I've got some more to say about that. Just one second.
All right. Yeah, we've been working on. Well, we've done some Hacktoberfest things in the past, and trying to get people in the repos, and you know, this has always been a struggle for our groups. I'm talking about DevaWorm as well as our group, and you know, we have different like open source software packages from GSOC and things like that to try to get people involved. And it's been a struggle to try to find a way to do that. And I think these hackathons are nice. We didn't have time to do this year to propose a project. But I think if people are interested in doing a project, more generally getting some experience, this is the way to go, to just register for one of these events. I guess in this case, BrainHack. And then maybe next year we can sponsor something through our own group. But yeah, and I know that Stefan Dvoretsky, who is, used to attend our meetings regularly, he's doing some things with EEG, and he might actually have a project at BrainHack. I don't know what he's doing currently with that, but that might be something to check out, or some of these other modeling packages that people are doing. So yeah, I think that's good. Definitely, let's follow up on that, and if you have. If people want to, kind of talk about it, if they want to get involved in that and share their experiences, that would be great. If you want to do it in a meeting, that would be great as well. We have anything else for today? Well, I just wanted to go over a couple things in the Slack before we go. And let me just do this real quick. And we didn't even do the Slack because we did it the reverse today, but I think we got a lot. So the brain organoids channel has been active. We have a number of different papers in there. We have this paper, A Matter of Space and Time, Emerging Rules of Disease-Associated Proteins and Neural Development. That's a nice review that came out this week. We have a couple of different papers on brain organoids developing optic vessels and responding to light. We have this neurogliovasculature portal, which Morgan mentioned previously, which is where, they're trying to build they're trying to characterize these other parts of the nervous system with glia and vasculature, the things that really kind of make a brain biological. But they've got this in the Blue Brain project. So this is something that is, I don't know if you can use the data. I mean, there are different ways you can reuse data. One of the ways is to get numeric values and just plot them out. Another way, of course, is to sort of find ways to use these models creatively. And so I don't know, people might have ideas for this that are beyond what I'm thinking, and please mention that. So there's a lot of stuff in the Brain Organoids channel. Another channel that...
Oh. Yeah, I was just going to add that, that the Montreal Concordia biomaking space director has been in touch and is really interested in working with us on the organoids.
Oh,
and
great.
Has said that I got him Katerina's paper and is already got all the parts, so he's like, I'm fabbing out the boards for the electrophysiological platform. And this is it was nice, or it was interesting also because I asked about another Concordia group that's focused on synthetic biology, and he said, well, yeah, I've met that person and I've seen their lab. The only certain PhD students and postdocs can access that lab. And I was like, yeah, again, I've been in community biospaces before, and I understand why you gotta have, it's making the right procedures is a challenge. But and a particular challenge in this case, right? Because again, like we've talked about, we're hoping for a bioreactor that maintains a cell culture for over a year.
Okay.
Right. It's like if you want an organoid that is a year old, you need to you can't bend time. And so this is quite a project. But I take it from their proactive moving ahead with things, that they really are interested in this, and it lights a fire under me to talk more with Katerina and her group at UCSD, Santa Cruz Brain Engineers, and see what else we can learn from protocols and bioreactors, and whether how much they are interested in kind of hosting organoids for us, or sharing protocols and additional information and hosting it from Montreal.
Great. Yeah, that's great.
Really, really, exciting, and so just want to say that.
Well, I'm glad the progress is being made on that front.
Yeah. And just the last bit about very soon. Okay. And just NeuroHack is a really interesting. It is sort of a hackathon, but it's definitely it's more a data hackathon, and a physiological, molecular, cellular data hackathon that is focused on dementia. Is an outgrowth of the Demon Network, which is a dementia focused research network. And I think it's more likely that like papers will come from NeuroHack, but that kind of work that's coming will be products of this hackathon, and interesting research collaborations, but I think more importantly, these will be like multi-scale examples of data processing.
Okay, yeah, that's great. Okay, one last thing I wanted to say before we go, and I wanted to go over the ethics channel. So this was Jesse, and he had to leave, but I think he knows about this, but some of everyone else knows. And Aaron, Angela, and Krishna were tagged in this. Timnit Gebru is a new research group that she founded after leaving Google, and so now this is a new research group that she's got, and you can investigate it. We mentioned this before. Read this article on Bloomberg News, or this is the DARE Institute. So this is the website, Distributed AI Research. So this is, again, it's a space for independent, community-rooted AI research, free from big tech's pervasive influence. So it'll be interesting to see what they do here in this space. And then there's this 100 Brilliant Women in AI Ethics video that Jesse posted as well. And then Aaron posted something on the sort of the energetic footprint of digitalization. So digitalization has become very prevalent in the last 15 years. How is it being powered? Is it being powered with renewables, or is there like a huge energy footprint? People have debated this, but it's something that is we should think about. So that's all I wanted to cover in that. I think you can go through the Slack and get some of the other things. There's stuff in the VR channel, for example, that I didn't go over, and I know Morgan was putting some things in there, so maybe next week we can talk about that channel more. So I think that's good for this week. Thanks for everyone for participating, and I think we had a really good conversation. I can share the shared notes documents with you if you ask, where they're in the channel, some of the channels, the Neuromatch channel, the Brain Organoids channel, and that sort of thing. So everyone have a good week. Talk to you in Slack. Take care.